Fingerprints are the most infallible means of identification. A forensic scientist who visits a scene of crime tries to procure as many fingerprints as possible. Fingerprints may be classified into three categories. The first are the inked prints, which are visible. You apply ink to your fingertip and impinge it on a surface. The print which we get is the inked print. The second are the latent fingerprints, which are invisible. They are formed by deposition of sweat on the surface with which the finger comes into contact. Since sweat is colorless, these prints are hidden, and that's why they are named as the latent fingerprints. The third variety is called plastic prints. They are partially visible. If one presses his or her finger on soap or on a freshly painted surface, a partially visible print is obtained. Blood prints normally come into this category. They are observed in cases where a person tries to stab the victim. They are found on the crime scene. They are found on the weapon. They may belong to both the accused as well as the victim. If the print is partially clear and we classify it as a plastic print, there is no need to go for further development. But experience shows that if the finger touches a surface again and yet again, then the intensity of the fingerprint goes down. And there is a need to develop such fingerprints. The development of blood fingerprints is based on the premise that there are two broad chemical characteristics of blood. The first is the red blood cells and the second is the serum or the protein content. Depending upon the type of blood which is found on the weapon, on the victim's body or on the walls, one has to choose the broad method, whether he wants to tag the blood serum or whether he wants to tag the red blood cells. Then those undeveloped fingerprints are treated with chemicals which give color derivatives with either of these two broad categories of chemicals which are present in blood. Phenolphthalein and malachite green are two chemicals which react with the red blood cells or the heme component, and they give color derivatives. You can have a dilute solution of these reagents. Even benzidine serves this purpose, but of late benzidine is suspected to be carcinogenic in nature, and therefore the forensic scientists normally avoid using it. Anyway, these chemicals react with the heme component of blood and they develop clear and sharp fingerprints on a large number of surfaces, both porous and non-porous. On the other hand, amido black and crystal violet are two very good reagents which tag the serum component of blood. They become attached to them and they impart their color to them. Even acridine orange may come in this category. So as usual, we take dilute solutions of these reagents and spray them on the weak blood fingerprint. The color developed is very sharp, very clear, very intense. And despite the fact that the original fingerprint is embedded beneath a layer of blood, we can still develop it and produce it in the court of law. There have been many cases which have been solved by enhancing the fingerprint buried under a layer of blood or buried under multiple layers of blood also, and with the aid of these reagents we can produce good results.
Dear students, after studying this module, you'll be able to know the significance of developing fingerprints on blood-laden evidence in crime cases. Then you'll come to understand the broad classification of reagents used for detecting fingerprints on blood smeared surfaces. You'll also learn detection of blood fingerprints with heme reacting and protein reacting chemicals. There are a host of chemical techniques for detecting latent fingerprints on different surfaces. However, if the surface happens to be stained by blood, as is common at crime scenes, then special procedures have to be adopted to lift the prints. Bloodstains are often found on weapons, victims' bodies, and other articles removed from the scene of crime. A scrutiny of these items may also reveal bloody fingerprints. Such fingerprints are generally visible since the ridges become stained with the red color of blood. However, if a blood coated finger touches a surface repeatedly, the immersion gets weaker and weaker. Under these circumstances, it is pertinent to enhance the impression with the aid of a suitable reagent so as to make the ridge detail clearer and sharper. The chemical reagents which enhance the blood fingerprints often tend to destroy or weaken the enzymes of forensic interest. Therefore, prior to fingerprint detection, blood samples from the impression should be collected for serological examination. Let us have a detailed look on blood smeared fingerprints. When fingerprints impinged on a blood stained article are to be detected, the method of choice depends on the nature of the surface and, more importantly, on the state of the coagulated blood. On coagulation, the serum content of blood separates out from the cellular content. The blood cells usually form a solid red mass which is surrounded by a layer of straw colored serum fraction. The serum proportion is rich in soluble proteins. The blood cell fraction is rich in heme. There are two broad methods for detecting blood fingerprints. If the finger touches predominantly the red blood cells on the surface of the crime scene evidence, the print may be developed by the application of a reagent that reacts with heme content of coagulated blood. If, on the other hand, the finger comes into contact with that portion of crime scene evidence which is coated with serum, the print may be developed by the application of a reagent that reacts with the protein content of coagulated blood. Now, let us focus on detection of blood fingerprints with heme reacting reagents. The heme moiety of the hemoglobin catalyzes the oxidation of a number of organic reagents. The oxidized reaction product is normally colored and therefore rendered visibility to fingerprint. Prior to 1974, benzidine was the most popular heme reacting chemical used for detecting blood fingerprints. Subsequently, however, benzidine was found to be carcinogenic and the fear of occupational exposure prompted the health authorities to ban its use as a fingerprint reagent. The following heme reacting reagents have proved useful and safe for detection of blood fingerprint. A. 3355 tetramethylbenzidine. The reagent 3355 tetramethylbenzidine, the structure of which is shown in the given figure, is a non-toxic and very effective in detecting blood finger marks. A buffer solution containing 10% by weight of sodium acetate and 80% by volume of glacial acetic acid is prepared. 200 ml of this solution, 2 gram of 3355 tetramethylbenzidine reagent is added. This is designated as solution A. A mixture of colloidal 30 ml, ethanol 15 ml, and diethyl ether 120 ml is separately prepared. This is designated as solution B. The working solution is prepared by dissolving 6 ml of solution A and 0.5 gram sodium perborate in 120 ml of solution B. The surface is impinged 2 to 3 times with the working solution from a distance of about 6 inches. Using a spray gun, one variety of which is shown in figure, on drying it at room temperature, the finger marks are rendered visible. Another reagent is phenolphthalein. A mixture of phenolphthalein, potassium hydroxide, and powdered zinc is placed in distilled water. The contents are refluxed for 2 to 3 hours until the formulation becomes colorless. This solution is kept in a dark colored bottle and care is taken that it remains in contact with zinc powder. Immediately before the fingerprints are to be developed, 20 ml of this solution is mixed with 80 ml of ethanol and 0.5 ml of 3% hydrogen peroxide. The test solution is applied to the article containing blood prints. On drying, the fingerprints become visible. Then, another reagent is leucomalachite green, an acidic solution of leucomalachite green reagent, the structure of which is displayed in the given figure, is quite effective in developing blood fingerprints. To a mixture of diethyl ether and glacial acetic acid, leucomalachite green and hydrogen peroxide are added. This working solution is sprayed onto the surface impinged with blood finger marks. On drying, the fingerprints are visualized. Now, let us see detection of blood fingerprints with protein reacting reagents. A good number of organic derivatives bind to the protein moiety in the blood to yield colored complexes. However, blood proteins are soluble in water. Therefore, before the article under examination is dipped in the test solution, it is baked in an oven for appropriate time, depending on the reagent being used. As a result, the proteins get denatured and become fixed on the surface. Therefore, the item is sprayed with the test solution of the reagent. The following protein reacting reagents have proved efficient in detecting blood fingerprints. Number 1, amido black. While using amido black reagent, the structure of which is set out in figure, three solutions need to be prepared. Solution A contains amido black dissolved in mixture of methanol and glacial acetic acid. Solution B is mixture of glacial acetic acid and methanol. Solution C also contains a mixture of glacial acetic acid and methanol but in a different proportion. The surface to be examined is baked in an oven at 100 degrees Celsius for about 30 minutes. It is then immersed successively in solution A and B. Finally, it is washed with solution C. The surface is allowed to dry naturally, whereupon the fingerprints are rendered visible. A representative blood fingerprint developed by amido black reagent is shown in the given figure. Then another reagent is crystal violet. The working solution is prepared by dissolving 0.1 gram crystal violet in 100 ml distilled water. The pH of the solution is adjusted to nearly 8 by adding a few drops of 1:1 aqueous ammonia. The article to be examined is heated to about 100 degrees Celsius for about 30 minutes. It is then sprayed with the test solution. After 2 to 3 minutes, it is rinsed with distilled water. On drying, the finger marks are rendered visible. A sample blood fingerprint developed by crystal violet reagent is shown in the given figure. Then, number 3 is Co-Massey Brilliant Blue R. Two solutions are required for developing blood fingerprints with Co-Massey blue reagent. The staining solution is prepared by dissolving 0.2 gram Co-Massey Brilliant Blue R 250 dye in a mixture of glacial acetic acid and methanol. The destaining solution contains all the ingredients of staining solution except the dye. The article to be examined baked at 100 degrees Celsius for 30 minutes. It is then immersed in staining solution for 3 minutes and subsequently rinsed with the destaining solution. On drying, the finger marks are rendered visible. Co-Massey blue reagent may also be used in concert with cyanoacrylate method of fingerprint detection. The cyanoacrylate method is based on the principle that when alkali 2 cyanoacrylate is allowed to vaporize, it undergoes polymerization. The polymerized ester has a tendency to get absorbed on the sweat residue. imparting a white color to the ridge pattern. The color contrast may be improved by various post-treatment methods. The surface impinged with blood prints is first baked in an oven for 5 minutes. It is then suspended from the roof of a cabinet in which is placed a china dish containing cyanoacrylate ester. The item is exposed to the vapors for about 2 hours until whitish colored fingerprint pattern develops. Thereafter, it is successively treated with the Comassi Brilliant Blue R staining and destaining solution. On drying, fingerprints appear. Another reagent is Crowell's reagent. Crowell's reagent is prepared by dissolving a mixture of Comassi Brilliant Blue R dye and crocin scarlet in glacial acetic acid, trichloroacetic acid, and distilled water. After the article is baked at 100 degrees Celsius for 30 minutes, it is immersed in the test solution for about 5 minutes with constant agitation. It is then repeatedly washed with water until the test solution background coloration disappears. The surface is then allowed to dry, upon which the fingerprints appear. Another reagent is ninhydrin. Ninhydrin technique has traditionally been the most popular one for processing latent fingerprints on porous, absorbent surfaces like paper, cardboard, and wood. The method relies on the reaction of ninhydrin, the, st- the structure of which is depicted in the given figure, with amino acid content of fingerprint residue. The reaction produces a purple colored compound called Ruhmann's purple, which becomes deposited along the ridges, making the latent prints visible. A zero point 0.2 to 1.5% solution of ninhydrin in Freon 113 is sprayed on the surface containing the finger mark from a distance of about 6 inches. After the solvent evaporates, the solution is resprayed. The surface is then heated to about 80 degrees Celsius without allowing it to come in contact with the heat source. Better results are obtained by steaming the article for optimum development of fingerprints occurs at the relative humidity of 65 to 80%. Ninhydrin method makes it possible to develop fingerprints that are many years old. However, since the concentration of amino acid in perspiration is usually quite low, the developed prints normally do not show a sharp contrast. In order to solve this problem, the developed prints are post treated with a group 12 metal salt, generally zinc chloride. This interaction converts the non-fluorescent Ruhmann's purple into a luminescent complex, the structure of which is depicted in the given figure. The fingerprints developed by ninhydrin zinc chloride methods become highly fluorescent under argon laser light. This procedure may be extended for developing bloody fingerprints. The blood protein is denatured and fixed on the surface by baking the sample in an oven at 100 degrees Celsius for about 5 minutes. Thereafter, the prints are sprayed with ninhydrin, followed by zinc chloride treatment, and then examined under laser. Dear students, now let us summarize this chapter. So, in this chapter, we have learned that blood fingerprints are often found at violent crime scenes. These may be detected by a legion of chemical methods. The detection methods are broadly classified into two categories: those which tag the heme content of blood smeared crime scene evidence and those which tag the protein content of coated blood. Apart from that, we have learned that, however, no single technique has universal application under all circumstances for the development of blood prints. And lastly, we have come to know that research efforts have focused on methodologies that may be applied to difficult and unusual surfaces. The method choice thus varies from case to case.